Hello and welcome to Medical Dialogues. I'm Anshika Mishra. Today we'll talk about research produces a new antibiotic-resistant to bacterial resistance. Researchers at the University of Illinois, Chicago and Harvard University have created an antibiotic that could provide medicine with a fresh tool to combat drug-resistant bacteria and their associated diseases. Published in Nature Chemical Biology, the antibiotic Cresomycin effectively suppresses pathogenic bacteria that have become resistant to many commonly prescribed antimicrobial drugs. The UIC scientists focused on how many antibiotics interact with a common cellular target, the ribosome, and how drug-resistant bacteria modify their ribosomes to defend themselves. The critical insights into cellular mechanisms and structure aided Harvard researchers in designing and synthesizing new drugs, including the development of new antibiotic. More than half of all antibiotics inhibit growth of pathogenic bacteria by interfering with their protein biosynthesis, a complex process catalyzed by the ribosome. Antibiotics bind to bacterial ribosome and disrupt this protein manufacturing process, causing bacterial invaders to die. But many bacterial species evolved simple defenses against this attack. In one's defense, they interfere with antibiotic activity by adding a single methyl group of 1 carbon and 3 hydrogen atoms to their ribosomes.
Are you also aspiring to excel in medical journalism and delve into the intricacies of health and medical reporting? Explore our comprehensive course on medical and health journalism available at the Medical Dialogues Academy. Learn from industry experts who possesses in-depth knowledge and experience in this field. For further details, scan the QR code provided or access the link in the description box below.
By using a method called X-ray crystallography to visualize drug-resistant ribosome with nearly atomic precision, scientists discovered 2 defensive tactics. The methyl group they found physically blocks the binding site, but it also changes the shape of the ribosome's inner guts, further disrupting antibiotic activity. By determining the actual structure of antibiotics interacting with 2 types of drug-resistant ribosomes, we saw what could not have been predicted by the available structural data or by computer modeling, said Yuri Polikonov, Associate Professor of Biological Sciences at UIC and colleagues at Harvard. Cresomycin is synthetic. It's pre-organized to avoid the methyl group interference and attach strongly to ribosomes, disrupting their function. This process involves locking the drug into a shape that is pre-optimized to bind to the ribosome, which helps it get around bacterial defenses. It simply binds to the ribosome and acts as if it doesn't care whether there was a methylation or not, Polikonov said. It overcomes several most common types of drug resistance easily. Without these structures, we would be blind in terms of how these drugs bind and act upon modified drug-resistant ribosomes. The structure that we determined provided fundamental insights into the molecular and mechanisms that allow these drugs to evade the resistance, concluded Polikonov. Thank you for watching. Stay tuned to Medical Dialogues for more updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.